Yes, thank you for the nice introduction, and I want to thank Evosep for giving me the opportunity to talk about my work here. And thank you to all who I assume are from all over the world. And good morning, good afternoon, or good evening to you. And so today I'll be talking about some of my work where we utilize ex vivo organ extracts from rats for identifying drug targets. And as I'm sure many of you can appreciate, proteins are the primary targets of most drugs. And even the best designed drugs can have a lot of off targets, which can be difficult to predict. And identifying some of these off targets or targets can actually perhaps help us elucidate the molecular mechanism of action for some of these compounds, but it could also help us design better drugs and perhaps repurpose existing drugs for different clinical diseases. And this is particularly challenging in the cancer research field, where it has been suggested that perhaps um, the common effect for most cancer drugs is through their unknown off targets or unknown mechanism of action. And so this particularly highlights the challenge in being able to identify the proper targets of different drugs in vivo. So why is that? Well, when we think of the way that most drugs are commonly designed, they are within a very narrow scope. And what I mean by that is you normally have your protein of interest, which you're trying to develop your drug for, and you will screen a large number of compounds until you find your lead compound, which can be further optimized to increase potency or bioavailability, for example. However, this approach typically fails to account for a lot of the cellular heterogeneity in which these proteins exist. And what I mean by that is these proteins can be modified with different modifications. They can be within the different parts of the cells in different organelles. And as we know now that many proteins exist in different protein complexes and form protein-proteins interactions. And all this is very dynamic. And this can be a bit difficult to take into account when you're designing a drug with, on a purified protein, for example. So there are many techniques out there which attempt to elucidate targets of different compounds. However, you're typically limited by the throughput at which you can do these techniques. And so an ideal technique, you will be able to screen a large number of proteins against a large number of compounds and rapidly determine which ones are targets. However, there is no, I would say, best technique. I think all different methods and techniques have their advantages and disadvantages. But being a protein researchers, we want to focus on techniques which um, work on the protein level because that is also where most of these drugs are working. And so it really is a compromise you have to make on the sensitivity of the technique, the depth, by which I mean how many proteins you can screen, as well as the throughput. And the technique that I found to be the best compromise for all this is based on thermal protein profiling or cellular thermal shift assay. And the advantages of this is that I've been really able to scale this up to perform it in a 96-well play format and basically perform the whole workflow in a 96-well play format, which allows you to do screen a large number of compounds and perform a large number of experiments in a relatively high throughput manner. And given the developments in some of the next generation mass spectrometers, as well as the Looker chromatographers, which are coupled to this, it really opens up the avenue to do this in a reproducible manner. And I'll talk a little bit about that in detail as well. For those who are not familiar, thermal protein profiling is a technique that's been around for a while, but a couple of years ago is demonstrated to perform this on a whole proteome scale. And the concept of this technique is if a protein is interacting with a small molecule or a ligand of interest, that can change the melting point. And the melting point is basically the temperature at which 50% of the protein is soluble. So, and if a protein interacts with a drug, that can change. It can either go higher or lower. And and if we remove the insoluble proteins after hitting these proteins, we can annotate which proteins could be targets of some of these drugs. Typically, this is done by measuring the solution or the protein at different temperatures and generating a melting curve from which you can deduce the melting point. However, we're not really interested in the melting curve per se. We want to just see whether or not interaction with the drug changes the protein solubility. So the approach that I'm using is basically integrating the melting curve over a couple of temperatures. And what I mean by that is basically heating the solution at different temperatures after it's been treated with a drug, and pulling the soluble fraction of those of the heating. And by measuring the protein abundance after that, we can deduce which could be potential targets of these drugs. And I'm not the first one to do this. This technique has been around for a while, and there's different variations of it. But the best strategy we found to utilize is basically coupling this with data independent analysis to basically increase quantitation as well as the throughput. And so here I have an example of HepG2 lysate treated with staurosporin, which is a non-specific kinase inhibitor. And when we perform this experiment, we can see that we really see a lot of the kinases we expect to see when the lysates are treated with this non-specific inhibitor. And so I basically extended this technique to utilize them on rat organs, specifically rat organs, different organs extracted from live rats, and from which we extract soluble proteins. And the advantages of using rats is that first, they're a common model system to perform preclinical toxicology studies. And second, we can obtain a large number of starting material and protein amounts from these organs. And so typically with these assays, from my experience, you need at least 100 to 200 micrograms per replicate. And if you're screening a large number of drugs and you're doing a large number of replicates, which I recommend around 4 or 5, you're going to quickly run through milligrams of material. So these organ extracts serve as a way to obtain a large amount of material, as well as we can screen different proteomes from these organ extracts, which allows us to perform these experiments across a variety of drugs and conditions. But I've also coupled this with 2 human cell lines, HeLa and HepG2. And the basic workflow, as I mentioned before, is you aliquot the lysates in a plate-based format, which you treat with different drugs. And you heat the extracts after treatment with the drugs, and you remove the insoluble proteins. And then the sample prep and the trypsin digestion is performed in the plate directly using protein aggregation capture or SP3 type method, from which the resulting tryptic peptides are loaded directly onto the Evosep. Again, everything is done in a 96 well plate format, and the resulting samples are analyzed by Orbitrap Explorer's mass spectrometer running in data independent acquisition mode coupled to an Evosep 1 method. I've been using the 30 samples per day method, the 44 minutes per run, to be like the best balance of depth and quantitation. And furthermore, we're analyzing everything using library-free DIA analysis or direct DIA, and I found Diane to work the best for that. And as a result, we can cover a large number of protein groups, typically over 9,000 protein groups if you combine all the different organs and cell lines. So that's pretty high coverage in terms of proteome depth. And just to give an example, here is muscle extracts treated from rats, treated with staurosporin. And we can see a lot of the kinases we expect to see when the lysate is treated with staurosporin, including this off-target, which has been known to result in adverse effects for some of these kinase drugs. And so we tested this across different organs, and generally we can see a response. It might seem like from this data set there's a lot of targets, but I should just mention that there is no, we didn't correct for multiple hypotheses, and this was just a preliminary test to determine whether or not the lysates were active, and we see that they are. And furthermore, we can see that the liver extracts are also active, which is good because liver is also where a lot of drugs are metabolized. And we can actually see that if the drug is incubated for too long with the liver extracts, that the drug is metabolized rather quickly. And this supports previous literature on storing, where rats were injected with the drug, and it was observed that the drug is rapidly metabolized within an hour. So this was a nice way to confirm that the extracts are active and they can be used for these type of experiments. So I've been performing these experiments with around 23 different drugs, some at different concentrations, and they can be kind of separated into either cancer drugs or metabolic drugs, roughly speaking. And as a confirmation, I have two drugs which target PTPN11, which is a tyrosine phosphatase, and these two drugs actually are in clinical trials right now. And we can clearly see when, in this case, the HeLa lysate is treated with these drugs, you can clearly see that the protein is stabilized, which we can measure in abundance here. And same goes for another cancer drug, which is cobimetinib, which is designed specifically to target MAP kinase 2K1 here. And you can see that it is stabilized by this drug, or when the HeLa lysates are treated. However, we actually also find that sometimes the engagement can be very context dependent. And this is just to highlight if we perform a volcano plot, we can see this target of cobimetinib again. Yes, as I mentioned that it can be very context dependent. In this case, in the rat muscle cells, we don't see as high an engagement as we do in HeLa cells for these two PTPN11 inhibitors. However, for the MAP2K1 kinase inhibitor, we can clearly see that it is actually engaging the protein. So it seems like context matter, and which also supports our strategy of utilizing different cellular contexts and organs. And again, if we visualize this using a volcano plot, we can clearly see that it is regulated. And in this case, the strategy we're using is to use all of the experiments as a control. And the reason for that is that you can get a lot of variability in the measurement, especially if the protein is low abundant. And when we're measuring melting point changes, we're not necessarily measuring protein abundance or protein expression changes, which you might typically associate with fold changes for proteins. We're measuring the amount that is soluble after temperature treatment for these different drugs. And the difference in change might not necessarily associate with how well a protein is engaging its target. And so we need very high accurate measurements and a good statistical power. And to do that, we're basically using all of the data set as a control. But at the same time, we do not want to use drugs which might have similar targets. So the way to control for that is something that Marie, who is a postdoc in Lars Jensen's group here at Center for Protein Research, strategy she came up with, which combines known targets from the drug bank database and using the structural features of the compounds and associating which compounds are too structurally similar to group together in a control, which is known as a Tanimoto score. And to give an example of that, for example, I have here these two compounds, RMC4550 and CHIP99, which are both designed to inhibit PTPN11. As you can see, they share a common target. So if we were looking for targets for this compound, when using all the other compounds as control, CHIP99 will be excluded from that. And that makes sense since they target the same protein and they're also structurally somewhat similar. And so we did that for all the compounds we're testing as a way to control for some of the shared common targets, which might be shared amongst different drug targets or different drugs, sorry. And so right now, I've actually performed the experiments for around 23 different drugs across all the different extracts and cell lines. So now the challenge really is in the data analysis, and that's where we're at right now. We have some exciting targets that we're doing some independent validation. So I'm waiting for maybe some more results on that so we can share that in a publication or a preprint, hopefully. But generally, using the strategy of organ extract seems to be a nice way to perform these experiments, and combining that with place-based assays and And place-based sample throughput is a good way, I think, to do these experiments in a relatively high throughput manner. Of course, this is really enabled by some of the advances in mass spectrometers, as well as the liquid chromatography systems which are coupled to it. So right now we're not really limited by the LC specifically per se. We're actually limited, I think, in my opinion, the mass spectrometers, because we can run these analyses faster using, for example, the 21-minute method per sample method. However, it will require, however, what we see is if we use the faster methods, we see a large drop in the number of identification and quantification. So for now, I think 44 minutes is the way to go. And we also have the challenge initially of basically converting the rat targets to human orthologs. And this was also compounded by the fact that the rat proteome, at least on Uniprot, is not very well annotated. It's only around 8,000 high confident annotations compared to 18 or 20,000 for human and mice, respectively. So we're using an ensemble generated FASTA for that, and that seems to work really well. And yes, and with that, I want to thank you for listening, and I want to thank for all the people that are involved in this project. Jesper Olsen for guidance and giving me the resources to carry out this project. Marie, who is a postdoc in Lars's group, for help on the data analysis and the normalization strategy that we're utilizing for this. Nadia for converting the rat orthologs to humans, and also doing some further downstream analysis, some network analysis. And also Stasia, who from the Department of Neurosciences, for getting the rats and performing the excisions to get those organs. And with that, I'll take any questions you have, and thank you for listening. And also thank you again to Evosep for giving me an opportunity to present my work here. Thank you.